Dear students, continuing with the nucleic acids, in today's lesson, we will be learning about different types of nucleic acid. First one, RNA or ribonucleic acid. This is the nucleic acid which is vital or essential for the synthesis of various proteins and for gene regulation. The chemical structure of the RNA nucleotide, this is an RNA strand, and this is the chemistry behind the nucleotide strand. As we have already studied, an RNA nucleotide consists of three components. One is the ribose sugar, which is a pentose sugar. It contains one OH group at the second carbon, and because of the presence of OH group, RNA is highly reactive and less stable than DNA. The second component is the nitrogenous base. Attached to the carbon 1 of the sugar, RNA contains 4 bases. Attached to this carbon, it contains 4 bases. One is adenine, then guanine, cytosine, and uracil. The third component is the phosphate group attached to the fifth carbon, connecting to the third carbon of the next nucleotide or the next sugar molecule. We have the phosphate groups. So, the structure of RNA contains phosphodiester backbone, that is between the ribose sugar and phosphate. It is single-stranded, unlike DNA double helix. RNA is single-stranded. Directionality is 5' to 3'. Types of RNA. There are three main types of RNA. First one is mRNA or messenger RNA, tRNA or transfer RNA, rRNA or ribosomal RNA. Messenger RNA, it carries the genetic code for the synthesis of protein, and transfer RNA delivers this code to the ribosomes, and ribosomal RNA forms the ribosome structure and catalyzes the synthesis of proteins. So in detail, first one, messenger RNA, as we have already told you, that serves as the template for the synthesis of protein. The synthesis of protein starts from an mRNA. And in mRNA, if you can see, it consists of a code or codon. A codon is a group of 3 bases. Like if you see here, UGG, that is a code. AUG is another code. UUC is another code. And this group of 3 bases of mRNA are known as the codons. Now, RNA, as we know, have 4 bases, but code for each amino acid has only 3 bases. So, 4:3 possibilities are there. That means 64 different codons are available. But as we know, there are only 61 alpha amino acids. So, the remaining 3 are for coding the chain termination of the protein. Transfer RNA. The main function is transfers the specific amino acid codes to the protein synthesis, to the ribosomes for protein synthesis. And transfer RNA is otherwise known as sRNA because it is soluble in water and smallest RNA. Both start with S. Now each tRNA consists of a segment called anticodon. It is ACU, which is a sequence of again 3 amino acids complementary to the codon sequence. 3 ribonucleotide or amino acids complementary to the codon sequence. For example, on mRNA, if you have a codon sequence CUG, then the anticodon will be GAC or tRNA. Ribosomal RNA, it provides a binding site for on the ribosome for tRNA and mRNA during protein synthesis. And ribosomal RNA is the most abundant RNA, consisting of almost 80% of the total RNA. And it combines with the proton to form ribosomes. And these ribosomal RNA are found in both small and large subunit of the ribosome. Now let us move on to the next nucleic acid, which is the deoxyribonucleic acid or DNA. It is a high molecular weight polymer composed of deoxyribonucleotide. It serves as the primary genetic material in living organism, and it is responsible for the storage, replication and transmission of genetic information. So DNA is actually the hereditary material in almost all living organisms. Structure of DNA, it is almost similar to that of DNA. Difference is that ribose in RNA is replaced by deoxyribose in DNA, and uracil in RNA is replaced by thymine. And unlike RNA, DNA is consist of two strands closely held together by the hydrogen bonds between the phosphodiester backbone. And hydrogen bonds occurs between specific pairs of bases. Now, details of hydrogen bonding in DNA. We know DNA also contains 4 bases, nitrogenous bases, A, G, C, T, adenine, guanine and cytosine and thymine. As you can see in this figure, adenine forms hydrogen bonds with thymine, and guanine forms hydrogen bonds with cytosine. And as you can see, between adenine and thymine there are 2 hydrogen bonds. Between guanine and cytosine there are 3 hydrogen bonds. Watson and Crick model for DNA. This is the most famous model explaining the structure of DNA. It was proposed by Watson and Crick in 1953, and as we know, they propose it as a double helix structure like this. That means it consists of 2 deoxyribonucleotide strands. One is going downside and the other is going upside. So it is actually 2 anti-parallel polynucleotide strands which contain a sugar phosphate backbone and a base pair stacked inside. So the key feature of Watson and Crick model is double helix structure. As stated, they have anti-parallel strands and a sugar phosphate backbone. Coming to the nitrogenous base pairing, the interior, that means this portion, this arrow portion, consists of nitrogenous bases where adenine pairs with thymine using 2 hydrogen bonds, guanine pairs with cytosine using 3 hydrogen bonds. Purine pyrimidine pairing is there. That means the large purines, that is purines are A and G, adenine and guanine. It always pairs with small pyrimidines, thymine and cytosine. So the diameter of a helix will be 20 angstrom. And the dimension between base pairs, between two base pairs is 3.4 angstrom. The full turn of a helix like this from here to here, it is 34 angstrom. And the full turn consists of 10 base pairs. Now let us have a small comparison between RNA and DNA. These are the key points. Sugar in DNA is deoxyribose. In RNA, it is ribose. Strands, double-stranded, single-stranded. Bases, ATGC, here AUGC. Helix, double helix. No stable double helix in RNA. DNA is more stable than RNA. This is due to the presence of an OH group in RNA. I have already explained. Location is mainly in the nucleus for DNA, whereas the RNA is present in the nucleus and cytoplasm. Function, that of DNA is basically genetic storage, whereas for RNA it is protein synthesis and gene regulation. Types of DNA, there is only one type, whereas RNA has several different types which we have already discussed. Now, a little bit more detail on the function of nucleic acids. Basically, we study only two main functions. One is the replication and the other is protein synthesis. Coming to replication, the main function of DNA is replication. That is, it is the property of a molecule to produce another identical molecule. That means the DNA can automatically make an identical DNA molecule by itself. So, in the first step, the two strands of DNA separate partially. That is, in the presence of an enzyme helicase, the two strands separate and each strand acts as a template for the synthesis of a complementary strand. So this is the step 1. Two strands of the DNA separate partially and each of these strands act as a template for the synthesis of a complementary strand. And the enzyme which unwinds the DNA strand is called helicase. It breaks the hydrogen bond between the base pairs and forms a replication fork in this manner. Now, due to the unique specificity of base pairing, the newly synthesized complementary strand will be an exact replica of the original strand. As a result of the replication, two identical DNA molecules are formed, and in these identical DNA strands formed are known as the daughter strands. And in daughter strand, one of the strands comes from the parent and the other one is newly synthesized. And this synthesis is helped by the enzyme DNA polymerase 3. Now, protein synthesis, another function carried out mainly by RNA. Protein synthesis is controlled by DNA and it involves two steps, which is transcription and translation. So this is transcription. Transcription is the first step of protein synthesis, where a particular segment, a part of the DNA molecule, or what we call a gene is copied onto the messenger RNA. This is done in the nucleus with the help of an enzyme RNA polymerase. So, in the first step, which is the transcription, a particular part of the DNA known as the gene is copied onto a messenger RNA by enzyme RNA polymerase. And this formation of mRNA, which is complementary to the DNA and is having the same DNA sequence, is called transcription. And the RNA thus formed is called mRNA. And in this process, DNA transfers the genetic code, a particular portion, to mRNA by means of replication. And this mRNA serves as a template for assembling amino acids in the polypeptide chain towards the formation of a protein. After the formation of mRNA, the mRNA detaches from the DNA nucleus. DNA molecule leaves the nucleus and enters the cytoplasm, where it serves as the template for protein synthesis. The next step is translation. Translation is a process where ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins by decoding mRNA into specific polypeptide chain based on the genetic code. So the first step in translation is initiation. In the initiation step, the small subunit of the ribosome binds to the mRNA. Now there will be a start codon AUG. AUG is known as the start codon that is recognized. Then the initiated tRNA carrying a methionine binds to this mRNA. Now the larger subunits joins and forms the complete ribosome. Ribosome, in the second step, elongation, ribosomes mainly has 3 sites. The A site, which is the amino acid site, which contains the incoming tRNA. This is the incoming tRNA. The P site or peptidyl site, which contains the growing chain, and the exit site or E site where tRNA lives. So in the elongation step, the tRNA enters the A site of the ribosome. There the codon-anticodon pairing occurs, peptide bonds are formed, and the ribosome moves. Chain shift to the P site or the peptidyl site. Termination step, when the stop codon, that is these 3 codons UAA, UAG and UGA appears, in this portion no tRNA binds. So the polypeptide chain is released in the presence of stop codon, and the ribosome dissociates from the protein. So that is all I have to tell you about the nucleic acid. Study well. If you have any doubt, you can contact me.